All right, it looks like we're ready to begin, so let's get started. Hello again, and welcome to Biosurgeon's webinar series. It's great to meet you all online. So in today's session, we will be talking about CNS drug delivery and BBB crossing, and we got some really exciting insights to share with you. So before we jump in, let me quickly introduce our team here. A quick intro of myself. My name is Yuan Tian, and I am the Content Marketing Manager at Biosurgeon Boston Corporation. I'll be the moderator for today's session. And I'm also joined by two amazing speakers today, Dr. Frank An and Dr. Meiqi Zhang. So first up, it's my honor to introduce Dr. Frank An. So Dr. An is the Vice President of the Institute of Biologics, which is Biosurgeon's Antibody Discovery Division. His key responsibilities include overseeing Biosurgeon's internal and external collaborative antibody discovery programs covering oncology, autoimmune disorders, and rare diseases, and including those that have reached preclinical candidates or PCC, non-clinical studies, IND, and clinical trial stages. He is also in charge of portfolio strategies, target inception and target product profile conceptualization, as well as overall project management and implementation. So Frank earned his PhD in cellular and molecular biology from Brandeis University, followed by a postdoc fellowship in immunology at Dana-Farber Cancer Institute and Harvard Medical School. With over 20 years of experience in drug discovery across industry and academia, Frank took increasing responsibilities before joining Biosurgeon, including positions at Bicycle Therapeutics, Horizon Discovery, Millennium Pharmaceuticals, which is now Takeda, and Broad Institute of Harvard and MIT. So great to have you here with us, Frank. Thank you so much for joining us today. And with that, let's meet our next speaker. Again, it's my pleasure to introduce our second speaker today, Dr. Meiqi Zhang. So Dr. Zhang received her PhD from Peking University, and she first joined Biosurgeon as a research fellow, focusing on animal model validation and evaluation for about 2 years. She then transitioned into the role of product manager and now leads the metabolic and neuroscience department within Biosurgeon's biomass product division. So in this role, Dr. Zhang oversees the development of many disease models and target humanized animals covering metabolic diseases, neurological disorders, and rare diseases. And she manages the full product lifecycle from product initiation and research to product development, model validation, pharmacodynamic studies, and technical support. So currently, her work focuses on neuroscience with key efforts in developing blood-brain barrier penetrating models, as well as models for Alzheimer's disease, Parkinson's disease, ALS, neuromuscular disorders, and more. So thank you, Dr. Zhang, for being here. Really looking forward to your insights. All right, now that you've met our speakers, here's a quick overview of today's agenda. We will start with Dr. An, who will share about Biosurgeon's recent advances in BBB shuttle antibody development, and then Dr. Zhang will follow with the role of humanized mouse models in supporting the evaluation of BBB delivery strategies. So with that, before we begin, just a quick note, this webinar is being recorded and we will have a Q&A session at the end. So as you're listening, feel free to drop in your questions into the Q&A box. We will go through as many questions as we can after the presentations. All right, that's it. Without further delay, I will hand it over to Dr. Frank An.
Okay. Thanks, Yuan, for the kind introduction, and thanks to our audience for coming to our webinar. So today I'm going to talk about antibody discovery for the blood-brain barrier, or in short, BBB shuttles. My talk is going to be divided into two parts. First, I would like to give a brief introduction of our company and also our antibody discovery platform. And then we're going to talk about the BBB shuttle discovery on the targeting TFR1, transferrin receptor 1, or CD71, and also CD98. First is introduction of Biosurgeon. A lot of you must have heard our company, and our businesses are in two divisions. First division is of focus is first division is called antibody therapy. It focuses on antibody therapeutics under the brand Ren Biologics. This is our antibody discovery arm. Uh, and also we have a preclinical arm that provide CRO services, mouse strains, etc., branded under BioMice. So today I'm going to talk about focus on the antibody discovery for BBB shuttles, and my colleague Meiqi Zhang is going to talk to you about the animal models that supports the BBB shuttle research. And oh, by the way, I forgot to mention that we're actually a global company, and we company is headquartered in Beijing, and with Asia's largest animal facility in Jiangsu province, and we have over 100 people strong team in Boston, and we have business offices in Shanghai, West Coast in the U.S., and also Heidelberg in Germany. So our antibody discovery work, we have been focusing on these following modalities. So we have an antibody, monoclonal antibody platform, and we'll have a bispecific antibody platform. And from there, we'll have a bispecific ADC platform. Then also, we have been working on this uh, it's a very small. It's people call that a single domain antibody. Also, this has other names like a human VHH, but it's just the single VH domain that's the smallest binding units. Okay, so that's we'll have a platform around that. And we also have two other modalities that one is a TCR. These two modalities focuses on identification of either TCRs or antibodies that for intracellular targets. So we have TCR. You can discover TCRs that targets intracellular targets, or we can generate TCR mimic antibodies that recognize the peptide and the MHC complex that to target intracellular proteins. Additionally, we'll also work on some harder to target classes of molecules such as GPCRs. And all these modalities or platforms, okay, are predicated on using our fully human transgenic mice. And collectively, they are called Ren mice. So, and here's a brief introduction of the Ren mice, and it has it's a suite. It's a collection of a suite of purely human transgenic mice. The first one is called the Renmab. The first strain is called the Renmab. Ren in Chinese means human. MABs here stands for monoclonal antibodies. So in these mice, what we have done engineered was that we replaced the mouse heavy chain locus with the human heavy chain locus. And similarly, we also replaced the mouse kappa light chain locus with the entire human kappa light chain locus. So in situ, by in situ hybridization, and recombination. So in these mice, they carry the entire human heavy chain repertoire and the entire human light chain, kappa light chain repertoire. So these mice are genetic wise, they carry the most diverse collection of human antibody genes. So that's a RenMab. And then the next strain is called a RenLight. Okay. Light means a common light chain. And here is that in these mice, they, again, similar to the RenMab, we introduced the entire human heavy chain repertoire into these mice. But on the light chain side, we only introduce a single human light chain. So in these mice, all the antibodies carry the same or common light chain. The the purpose to engineer a strain like that is to allow the correct the pairing between light chain and heavy chains. Okay, so that can this is very useful to when you wanted to make bispecific or multispecific antibodies. And the last strain is called the nano. Okay, nano is refers to the product that is the VH binding domain, and people also call that the nanobodies, the VHH, single domain antibodies or heavy chain only antibodies. It means the same thing. That here is the we engineered the mice so the antibodies generated from these mice only carries this VH domain. Okay, so these are the three basic mouse strains under the mice. And we used these fully human transgenic mice, and one of the things we used them for was to generate over 1 million fully human antibody sequences, and also with the binding informations, and sometimes with some basic characterizations such as internalization activities. So these are fully annotated fully human antibody sequences that targets over 1,000 targets. So here is just give you a flavor of some of these targets we have worked on in oncology, inflammation, autoimmune diseases, neuroscience, etc. And also from those binders, we also developed some advanced molecules and worked together, either licensed or co-developed with our collaborators who put them into clinical trials. So here is some of the recent examples. We have 3 bispecific ADCs in phase 1 clinical trials. And we also here shows an anti-IGF-1R antibody involved in autoimmune disease called thyroid eye disease. And that this molecule entered a clinical trial in China earlier this year. And we have a bispecific molecule that targets PD-1 and CD40. And here we also contributed 2 components of this 4-component cancer vaccine that showed actually, superb activity in phase 1 trial against prostate cancer. So with that introduction of our antibody discovery programs, platforms, and I'm going to turn to the main topic of today, that is discovering BBB shuttles. So the first target is transferrin receptor 1, TFR1, also called CD71. So it is a protein that is highly expressed on the surface of endothelial cells, and its role is to transfer. Its normal physiology is to transfer iron, in the form of transferrin into tissues. So so that's its normal function, and it's actually clinically validated targets for blood-brain barrier penetration. And this diagram actually illustrates that this is an anti-A-beta antibody fused to anti-transferrin receptor TFR1 antibody, and that would target the TFR1 on the BBB barrier, and then translocate and bring the antibody into the brain so that the antibody could bind the A-beta plaques and clear them. So that's, and here is just a view of some lead molecules in the field that targets TFR1. JR141 was approved product for antibody replacement therapy, and the trontinemab is a new star for Alzheimer's disease from Roche. So really, this target gained prominence after Roche published their work that showed dramatic improvement in terms of distribution in the brain. Here is just anti-A beta antibody that showed a very restricted choroid plexus distribution in mouse brain. But when they fused this antibody to a anti-TFR1 moiety, and you can see that the distribution has become very evenly distributed in the mouse brain. And clinically, it's also made a huge difference. Here is their, A beta TFR1 bispecific antibody, trontinemab, and in clinical trials, you can see very quickly within 28 weeks that the A beta plaque dropped. This is centiloid in terms of centiloid, almost 100% clearance of plaques. But they also run trials before trontinemab with this antibody that does not have the anti-TFR1 moiety, and you can see 28 weeks point, there's only less than 20% improvement in terms of centiloid. So after they published this work, it generated a lot of interest in the field targeting TFR1, and so we set out also to make the anti-TFR1 antibodies with our nano fully transgenic strain. So here is our immunization. We scored several clones, and this shows the germline usage and the distribution of highly chain CDR3 amino acids lengths. So that just shows a decent, a very good diversity. And here is the species cross reaction, where we tried the binding transiently expressed human TFR1 and the monkey TFR1s. You can see some clones. Those red ones shows cross species reactivity against human and monkey, but these clones don't bind mouse TFR1. And here's some affinity information, and you can see there's a nice distribution, -8, -9 affinity for these clones. And we also tested, we don't want them to block the transferrin, normal transferrin receptor function. So and you can see here, for example, antibody 26, which is the red line, okay, did not block transferrin and the transferrin receptor association. And so that's what we want for these clones. And here is the data for in vivo characterization of our TFR1 clones. And here I'll just focus on the parenchyma fraction. And the positive control is this JR141, the approved anti-TFR1 molecule. And that is in the blue line. And we have, our antibodies in different colors, and but you can see that some of our clones, like the red one, okay, and this purple one, show the better brain penetration than the positive control. And here to show some staining in the brain that shows that the antibodies actually would co-localize with the neurons. So that's a co-localization study. And here is a further characterizations, and here we generated the monovalent anti-TFR1 molecules. Here's AB26, which is our lead molecule here. And you can see there's a nice dose response in the monovalent form, and the monovalent form worked better than the bivalent form. Okay, so this is well accepted in the field that the monovalent format would work better than the bivalent format for anti-TFR1s. And we also show that our, human VHH or single domain antibody are very amenable for engineering. And here we could put it at the C terminus and could put it at N terminus. and this data shows that essentially there's no difference in terms of BBB penetration. And also, one of the challenges for these single domain antibodies is actually their biophysical properties and the developability. So here we shows that it has very good biophysical properties in terms of different parameters. Here is aggregation tendency, so they show the very good lack of that, and good thermal properties. And here, this panel shows that actually it has a very good thermal stability. So we subjected the molecule, the 40 degrees for 14 days, which is this orange bar, compared to the day zero, its original status. And there's no change in terms of different in terms of the composition of this molecule. And also, after we obtained those antibodies, and a lot of the requests actually focused on antibody oligonucleotide conjugates. So a lot of people are working on RNA, small RNA inhibitors, and actually, for these applications, what the request was that they need DAR1, because for the PK reasons, DAR1, that means the antibody and oligos in a 1-to-1 ratio. So that's would allow them would allow the scientists to do the AOC applications. It's actually quite challenging very challenging to for this symmetric, by chimeric antibody framework to generate a DAR of 1. So we did engineering on the molecule, and so our molecule, would allow homogeneous and site-specific conjugation and for DAR1. Okay, and the chemistry, we chose a chemistry that is universal, is a cysteine and melamine reaction. So people would have complete freedom to operate. And here is some of the analytical data, and we piloted it with payload conjugation, like the VCMMAE. It's a small molecule payload, and we wanted to see DAR1 conjugation. And here's just some crude reaction mix after the conjugation reaction, and you can see there's 85%, 80% on different analytical columns. That's the purity, so that's very good. And we also, confirmed the identity of those molecules by mass spec. Then in vivo testing showed, for example, here we use here's the purple, that's AB26 with DAR1 engineering, and you can see it shows comparable BBB activity relative to its parental form. Okay, so in the last slide, actually, I'm going to just do a brief introduction about our targeting efforts on CD98. So CD98 is actually is a component of amino acid transporters, and so that's its normal physiological role. And it's also reported important for T-cell mediated immunity, and people also use that as a oncology targets. But recently, because it's high expression in brain endothelial cells, so people also wanted to use that as a BBB shuttle target. So here, this panel shows that here is the red one is the anti-CD98 positive report, a molecule reported in the literature from Jensen, and that's the BBB penetration profile over time. And the green one is our positive control benchmark molecule. this GR141 targeting CD71 or TFR1. And so you can see that these two targets have different kinetics and different exposure and among other differences. And so it's making this CD98 a really intriguing target for BBB the penetration. Just we're waiting for the first clinical evidence that about um, you know anti-CD98 as a BBB shuttle. I mean, when I say we, that means the community. And but barring that, so we worked we used the Ren Nano and the Ren Light strain and generated a series of anti-CD98 clones that actually showed superior brain exposure versus our benchmark molecules. Just due to the legal reasons, I cannot disclose the unpatented material data with you today. But please feel free to approach any biocytogen people, in particular our BD team, and with the CDA, we would be able to talk to you, present our work with our anti-CD98 clones. So that's is how we introduce these data to you. With that, just I'll stop here and thank you very much for your attention.
Thanks,
So
Frank.
I'm going to stop share and give the floor to my colleague.
And while we're waiting for Dr. Zhang to pull up her slides, feel free to go ahead and drop in any questions you already have for the first part of the presentation. We will have the Q&A session right after Dr. Zhang's presentation. So keep them coming.
Okay, thank you, Yuan. And hello everyone, and I'm Meiqi. I'm a product manager from Biocytogen Biomass department, and I'm a major in the development of humanized mice models or disease mice models. And today I will share animal models for the blood-brain barrier crossing, and these models maybe you're helpful to in the preclinical research. And our biomass product lines is the most comprehensive human targeted knocking mice or cell lines. And we have many products, including over 1,000 targeted humanized mice models, including the immune checkpoint humanized mice, cytokine and cytokine receptor humanized mice, and GPCR humanized mice. And we also have about 100 seriously immune deficiency models. And we also have many disease models in multiple areas such as the aging, the blood disease, the metabolic disease, and the neurological disease. And we also have over 700 gene knockout mouse models, and these models can be used for the target validation. And now we have over 4,000 mice models off the shelf, covering all the research series. And yes, we have many products. And today I will share with you these topics. First, I will give a brief introduction of the receptor-mediated transcytosis strategy. And second, I will mainly introduce the animal models for the blood-brain barrier crossing, including the humanized TFR1 mice, humanized 1970 heavy chain mice, and the third one is the IGF-1R humanized mice. And the last one is the summary. And we all know that blood-brain barrier is a highly specialized interface that takes control of entrance of the molecules. And it's said that more than 98% of the small molecular drugs and about 100% of large molecular drugs, including the antibodies, enzymes, or some nucleic acid drugs cannot cross the blood-brain barrier. So to address the restrictive nature of the blood-brain barrier, multiple strategies have been explored, and one of these is the receptor-mediated transcytosis. antibodies. This strategy is a cellular transporter that facilitates the movement of specific molecules across the blood-brain barrier. And the most famous target, which have also introduced by Dr. An, is the TFR1 and the CD19 heavy chain, these two molecular. And the third molecular is IGF-1R, which is recently being developed in the blood-brain barrier transportation. And so next, I will introduce the animal models for the blood-brain barrier crossing. And the first one is the humanized models for TFR1. This is the background of the TFR1, and Dr. An have also introduced TFR1 is the receptor of transferrin and functions in the iron transport through transcytosis mechanisms, and is highly expressed on the surface of endothelial cells in the blood-brain barrier. And both the Denali and Roche and many other companies have developed the transportation platform targeting the TFR1 for blood-brain barrier penetration, including some antibody drugs or some antibody oligonucleotide conjugates penetration. And the targeting strategy of the humanized TFR1 mice developed by Cytogen is the in situ genomic replacement. And we humanize the whole actual domain of the TFR1 encoded by the exons 4 to exons 19. And the transmembrane and intracellular domain was returned as the mouse sequence. And for this mice, we have do some mRNA expression or protein expression analysis. In the mRNA expression data, we can see that the mouse TFR1 mRNA was totally knocked out, and in the homozygous mice, only the human TFR1 mRNA was expressed, and this sequence was also confirmed by Sanger sequencing. And and we also do some protein expression analysis. First, we test the TFR1 expression in the bone marrow red cells. We can see that the mouse TFR1 was knocked out in the humanized mice, and only the human TFR1 protein can express in the humanized mice. And we also detected 3 male and 3 female mice, and the human TFR1 protein expression levels were basically consistent between the male and the female mice. And next, we detect protein expression of TFR1 in the brain endothelial cells by flow cytometry. And we can see that in the wild type mice, the mouse TFR1 protein expressed at about 25%. And in the homozygous humanized mice, the human TFR1 protein expressed at about 35%. And we think the expression levels in the wild type mice and the humanized mice remain consistent. And we also test the TFR1 expression in many other tissues, including the normal tissues or some reproductive tissues, such as the heart, liver, spleen line, kidney, and some many other reproductive tissues. And we can see that the TFR1 expressed in many tissues, and the expression profiles of the mouse TFR1 and the humanized TFR1 are very similar, which is also very similar to that in the humans. And next, we detect the healthy status of these mice. And first, we performed the leukocyte cell subpopulations analysis. And we want to see if humanization of TFR1 will change the overall frequency of the immune cells. We detected the spleen, blood, and lymph node. We detect many immune cell types, including the T cells, B cells, NK cells, DC cells, and some monocytes and macrophages. And we can see that in the humanized mice, all the immune cell types, the percentage of the immune cell types in the humanized mice is as similar as that in the wild type mice, which is the humanization of TFR1 doesn't change the overall frequency of the distribution of the immune cell types. And since TFR1 function is related with iron transport and related with the development of red blood cells, so if TFR1 doesn't function, the mice will have anemia. So we tested 20 parameters in the blood routine test, including the red blood cell count and 2 parameters in the red cell distribution, and also the reticulocytes parameters. And these parameters are mainly used in the assessment of anemia. We can see that in the humanized mice, these parameters in the humanized mice is similar to the values in the wild type mice, indicating that the humanized TFR1 mice don't have anemia. And beside the blood protein test, we also have tested 3 parameters in the iron homeostasis, including the serum iron, mouse ferritin, and mouse transferrin. And both the 3 parameters in the humanized mice is similar to that in the wild type mice. So from this data, we can see that humanized TFR1 can function well in these mice. And we also have tested 8 parameters in the blood biochemistry test, and also there don't have any abnormalities in these mice. And next, we do some applications of these mice, and this is an imaging method to see if the antibodies can enter the brain. We conjugate the TFR1 antibody with fluorescence, and after dosing, we take the whole brain under the imaging. From the data, we can see that the two TFR1 antibody, the signal intensity of the two antibody is higher than the isotype group, and the anti-TFR1 antibodies can enter into the brain. But actually, this is a low resolution method that can't accurately assess whether the drug is still on the blood vessels or has actually entered the brain parenchyma. So we also use another high resolution method, which is AOCs, to image the antibodies in the brain. And after the drug administration and the perfusion, the whole brain was harvested for the IVs imaging. And then this method can show the antibody distribution and 3D level using a light slide microscopy. So from this figure, we can see that the anti-TFR1 antibody is enriched in the blood vessels, but in the brain parenchyma, there is also some signals of the antibodies, and which can proving that TFR1 antibody can enter into the brain parenchyma. And we also have ELISA method to assess the kinetics of the antibodies. So here is the workflow. After the drug administration, we can collect the blood sample, and we can also collect CSF. And after the heart perfusion, we can collect brain for next detection. And usually we will take half of the brain to do the IHC staining, and another half of the brain in the ELISA detection. And the brain tissues were homogenized, and adding the dextrin and centrifuge to get 3 layers. And we usually take the top one and the middle layers for ELISA detection to get accurate antibody concentrations in the brain, not enriched in the blood vessels. And the concentration in the whole brain can be 5 to 6 times higher than that in the brain parenchyma. And this part has also been shown on the previous report. Dr. An have shown the data. And using this method, we also test the efficiency of some anti-TFR1 antibodies penetrating into the blood-brain barrier. And this is data provided by one of our client, and they developed anti-human TFR1 bispecific antibodies. And we can see that the brain serum ratio of this bispecific antibody is higher than the control group. And here is also another case developed by one of our client. They developed a series of antibodies with different affinities, and as the affinity decrease, the antibody concentrations in the brain parenchyma first they increase and then decrease. This is also consistent with the theoretical basis, because if the affinity of the TFR1 antibody is too strong, it will make it difficult to dissociate, causing it to be retained in the blood vessels. And finally, the brain parenchyma concentrations of the antibody may be very lower. And besides antibody drugs, humanized TFR1 mice can also be used in AAV penetration into the blood-brain barrier. And this is also a literature published by one of our clients. They developed an AAV capsid reprogrammed to bind human TFR1, and they delivered the AAV capsid to the blood-brain barrier. And this year, FDA has also approved this AAV capsid for safety research, just using our humanized TFR1 mice. Besides, TFR1 can penetrate the large molecular drugs to the brain. TFR1 can also be used for drugs delivery into the muscle. And here is a case study. We developed the TFR1 and the DMPK double humanized mice by biocytogen, and we can see that in the muscle tissues, the DMPK mRNA was decreased after the dosing of the antibody oligonucleotide conjugates drug. And here is also another publication published by one of our clients using TFR1 humanized mice in the drug delivery into the muscle. And this is the summary of the humanized mice models. Our humanized TFR1 mice, the knocking strategy of these mice is for genomic in situ replacement of the entire extracellular domain, and we keep the transmembrane and intracellular domain as a mouse sequence. And these mice, the phenotypes of these mice is normal. We test the blood biochemistry and hematology test, and we have serum iron test, we have birth data and growth curve. All these data showed that the humanized mice is normal. It doesn't have any abnormal phenotypes. And now we have tested monoclonal antibodies, bispecific antibodies, AOC drug, and AAV capsids penetrating into the blood-brain barrier or muscle. And for these mice, we also have crossed these mice with many other humanized mice models or some disease mice models. So we have a lot of products on the shelf. And next, I want to introduce humanized models for CD98 heavy chain developed by biocytogen. As Dr. An just mentioned, CD98 heavy chain is a rising star in the blood-brain barrier transport, and then they have also developed the transportation platform. And for this target, we also have humanized mice models. The gene targeting strategy is similar to that in the humanized TF1 mice, and which is also the in situ genomic replacement. And we also humanized the extracellular domain of the CD98 heavy chain, and we also keep the transmembrane and intracellular domain as a mouse sequence. And for this month, we also have mRNA expression data and protein expression data. In the mRNA expression data, we can see that the mouse CD98 heavy chain mRNA was knocked out, and in the humanized mice, only the human CD98 mRNA was detected, and this sequence was also confirmed by single sequencing. And for the protein detection, we first detect CD98 heavy chain expression in the TB cells, and we also detected its expression in the brain endothelial cells. From this data, we can see that in the brain endothelial cells, CD98 heavy chain is highly expressed in the brain. And we also have tested CD98 heavy chain expression in many other tissues, including heart, liver, spleen, lung, kidney, and brain. We can see that CD98 heavy chain is highly expressed in the brain and kidney. So one advantage of the CD98 heavy chain over the TF1 may be the peripheral delivery efficiency of the CD98 heavy chain antibody may be lower than the TF1 antibody. And here is also some data of this mice healthy status. First, there is also the analysis of leukocyte cell subpopulations. We can also see that humanized CD98 heavy chain doesn't change the overall frequency or distribution of the immune cell types. And here is also the complete blood count data and biochemical test. And in the humanized CD98 heavy chain mice, all the parameters shows the similar as in the wild type mice. And these mice don't have any abnormal phenotypes. And for this mice, we also have an PK study data. We use an anti-CD98 heavy chain antibody, and we can see that the brain serum ratio of the CD98 heavy chain antibody was higher than the control group. And the third one is the humanized models for the IGF-1R. IGF-1R is a tyrosine kinases receptor that bind with IGF-1, and its functions in promote cell growth, survival, or metabolisms. And recently, a company have developed a grapple DB platform using the IGF-1R for the blood-brain barrier penetration. So we also have this humanized mice. The targeting strategy of this mice is similar to the former two mice, and we also humanized the extracellular domain of the IGF-1R, and the transmembrane and intracellular domain of the IGF-1R was returned as a mouse sequence. And for this mice, we also have mRNA and protein expression analysis. This data was similar to the former two humanized mice. And this mice, we also have immunofluorescence analysis in the brain, and we can see that the IGF-1R is highly expressed in the brain macrovessels. And we also have a function assay of these mice. After adding the human IGF, the human IGF-1, it can activate the receptor, which is IGF-1R, and the phosphorylation of IGF-1R will increase. But if we add an antibody targeting the IGF-1R, the phosphorylation of this receptor will be blocked. So in this assay, we can see that the humanized IGF-1R can function well in these mice. And the last one is the summary part. So in the strategy of crossing blood-brain barrier, the receptor-mediated transcytosis is a mainly strategy. And in this strategy, the most famous target is the TFR1, CD98 heavy chain, and IGF-1R. And for TFR1 related mice models, we have developed a series mice, and most of them are on the shell. And for the CD98 heavy chain and IGF-1R humanized models, we also have developed a series of models. Yeah. And today this is all for my sharing. Thank you.
Thank you, Meiqi, for the great presentation. I think we already got quite a few questions in the Q&A box, so I'll take it from here as we move into the Q&A session. And to our audience, if you haven't submitted your questions yet, go ahead and do it now. We will try to get through as many as we can today. All right, so let's start with this first one here. So anti-TFR1 may kill reticulocyte and cause anemia. Did you test whether your anti-TFR1 nanobody causes anemia? This one goes to Frank.
Yeah, that's a very good question. And yes, that's a well-known side effect for targeting CD71 or transferrin receptor. Um, excuse me. So we have tested that. Because of the time limit, I did not show that. And we have various properties for different clones. What I can just suggest here is that please, contact our BD team. We can set up a time and go over some additional data and in particular regarding your question about the reticulocytes cytotoxicity.
Great, thanks, Frank. I think there's another question also for the antibody part. In addition to what you presented, do you continue generating additional antibody clones, or could this be tailored to meet specific project needs?
Yes, that's again another very good question. We have been continuing the clone discovery because it's a complicated situation where you need to have a very active, but very also has a good safety profile for these clones. So we are we keep improving our on those properties. So it's a continuous efforts for us to discover additional clones. Then again, please get in touch with our business development teams or anyone from Biocytogen. And that will eventually lead to us, the discovery team, and we can, talk about the new clones and the new properties, etc. with you.
Thanks, Frank. I think that's also covered another question here, like how to contact for additional information on the antibody programs. You can find almost all the contact information on our biosurgeon.com website, and you can also contact the BD of the RimBio sector to get more detailed information about those antibodies or our programs. Okay, here's another question. Do you know the distribution of the CD98 shuttle in other organs besides the brain?
Yes, CD98 is actually expressed in multiple places, and in addition to endothelial cells, and it's also expressed in a few immune cell types. The most prominent one would be T cells and B cells and macrophages. The placenta is another organ that in pregnant women that where CD98 is highly expressed. So, so it's not exclusive for endothelial cells, and none of those things are, the current, popular targets for BBB shuttles. So it's really it's you have to balance the brain transcytosis activity versus the potential safety concerns. But at least in terms of CD71, the clinical trials have demonstrated a I would say minimum an acceptable and well managed safety profile for these PBB shuttles. And for CD98, we're eagerly awaiting for the first clinical trial results to show us how to balance the transcytosis activity versus the potential side effect from this target.
Thanks, Frank, for the detailed answer. I think we can take one like humanized model question first. This one is for Meiqi. So after humanization, can human TFR1 function normally, and does it cross-react with mouse transferrin?
Oh yeah, humanized TF1 functions well in the humanized mice, and it's reported that it's reported that the TF1 homozygous knockout mice are embryonically lethal, showing anemia and some neurological defects. And our humanized mice can birth normal, and we have also tested many other phenotypes data, such as the hematology analysis data and the biochemistry data, and we also have some iron homeostasis related data in these mice, and all the results was similar to those in the wild type mice. So the humanized TF1 functions in these humanized mice, and also the mouse transferrin combined to the human TF1, and these mice don't have any abnormal phenotypes.
Great, thanks, Meiqi. And another question about the mouse model. Do you have validation data for the delivery of small nucleic acid drugs?
Yes, we have some data, and for the small nucleic acid drugs delivery into the brain, we have TFR1 and tau double humanized mice, and we have tested the siRNA delivery into the brain, and we have some data against the tau mRNA expression levels. And we also have delivered data into the muscle using double humanized mice, which is the TFR1 and the DMPK humanized mice. And we also have some knockdown data in our product menu. Yeah.
Great, thanks, Meiqi. I think there are two questions asking the same thing about the Alzheimer's disease model. I'll just read one because the other one is literally the same question. So you have mice that combine an Alzheimer's disease model like 5-time FAD with TFR1 humanization?
Yes, we have crossed the we have this double humanized mice by crossing TF1 humanized mice with the transgenic 5X FAD mice. And for this double humanized mice, we also have some validation data, including some phenotypic data such as the A beta deposition, astrocyte activation, or biomarker detection. We also have behavior tests such as water maze, and this strain is available now.
Great. Thanks, Meiqi.
Yeah.
Let's take one more antibody question. If you could recommend just one antibody product from your company, which would it be?
Sorry, just one antibody product or one of those targeted product and an anti-TFR1 or
I think either is fine
anti-CD90?
because they didn't specify just like one antibody
Okay.
product.
Okay. Well, since we're talking about the BBB shuttle, okay, I'll just say that for those targets, we have multiple clones. Okay, and we have multiple clones because they have different slightly different properties in terms of biological activities and also physical properties, etc. So we do have lead clones, and but we're also very mindful that different applications might be different. So when we talk to you, we will show you our lead clones, how they perform, but we'll also listen to your specific need for your project, and we're very adaptable and trying to work with you to give you some molecules that would fit your research need. So just very short is that we do have lead clones. Contact us, so we'll show you. But if these lead clones need further modifications, we'll be very open to doing that and working together with you.
Great. Thanks, Frank. It looks like we're getting close to the end of the session, so let's take one last question here. So in some clinical trials, anti-TFR1 treatments cause some side effects that has a concern to use it in patients. Do you have any observation in your animal models?
In animal models, we did not observe any acute, the side effect or discomfort of the animals. The lowering of reticulocytes numbers and other properties, yes, we did observe that, but that also varies clone by clone. Some clones showed a very good rebound of reticulocytes counts after a few days. So, clearly these things are not, restrictively expressed in only one region in the body. So there might be, other side effect, but anemia, reticulocytes are well-known things for targeting CD71. But however, this said, people actually, are doing different things trying to mitigate that risk, and there's a way to remove pretty much that toxicity altogether. It depends on the targets. It depends on the cargo you wanted it to do. So there's different ways to mitigate that.
Awesome. Thanks, Frank. Unfortunately, I think that's all the time we have for questions today. Before we wrap up, I want to give a big thank you to Dr. Ann and Dr. Zhang for sharing their insights. And of course, a huge thank you to everyone in the audience for engagement and questions. I hope you picked up some valuable takeaways from this session. That said, if you have any follow-up questions, or if you want to learn more about our BBB shuttle molecules or humanized mice, feel free to reach out through our website at biocellogen.com or connect with us on LinkedIn. We would be happy to continue the conversation. And in case you missed any part of today's session, this webinar will be available on demand. You will be able to request a recording directly on our website. And also, I saw several questions asking about the slides sharing. Also, again, if you want to have the slides, feel free to contact us on biocellogen.com or LinkedIn. We would be able to share the presentation slides afterwards. All right, I think that's all from us for today. Thanks again for joining. Have a great rest of the day and hope to see you at our next webinar. Thank you.
Thank you. Bye bye.
Okay.